Hey, Mr. P here. In this video, we're going to talk about cell structure and the function that the cell structures need to have in order to function correctly. This isn't a really in-depth dive into or intense dive into cellular structure, but for the purposes of this class and the purposes of this unit, this is kind of the depth that we need to understand. It's primarily a review of the content you've had in previous classes. And so this is just going to be kind of a once-over introductory video of cellular structure and function. So let's dive in. When we talk about cell structure and function, there's really two areas within the cell that we need to talk about. One of those being the plasma membrane and the other one being everything else. Basically everything within the cell membrane. And everything within the cell membrane or everything interior of the cell membrane would be kind of within the cytoplasm component. Okay? So two cellular domains, plasma membrane and cytoplasm. The membrane consists of a lot of different structures that helps to separate the cell from the external environment. The cell membrane really does two things. One, it serves as a boundary a boundary between the inside and outside of the cell. It also, because it's a boundary, serves to control the exchange of materials that are going to be swapped between the inside and outside of the cell. Okay, most of what is swapped or most of what is going to be moved from inside to outside or outside to inside is going to be moved through these channel proteins, which is one of the protein components of the plasma membrane. The plasma membrane consists primarily of phospholipids, and you can see those here in these little purple, these little purple structures. It's a phosphate head and two fatty acid tails. We talked about that in the last video. Well, now when we put those phospholipids together, you can see that they orient themselves into a sheet, and it's called a phospholipid bilayer or a plasma membrane or a cell membrane. Because the heads are hydrophilic and the tails are hydrophobic, the tails need to orient themselves interior, and the heads can be oriented out because it serves, again, as that boundary between the water on the outside of the cell and the water on the inside of the cell. Okay, so water is going to interact with the heads on the inside of the layer, or on the inside of the membrane, and the water is going to interact with the heads on the outside of the membrane. Again, we just said in our last video that this creates a zone of hydrophobicity, which is one of the most important parts about the cell membrane because it kind of eliminates the risk of certain things, certain polar molecules moving through the membrane when they shouldn't. So, because the phospholipids make the general bulk or the foundation of the membrane, it serves as anchor points or anchorage points for a lot of other proteins. Some of these proteins can be integral, meaning they span the entire membrane, or the entire width or thickness of the membrane. They are completely integrated into the membrane. There are other proteins that are peripheral, which you can see here, called a surface protein. That's a protein that is on the periphery. It can be either on the outside or inside, but it does not span through the entirety of the membrane. You have glycoproteins, which are proteins associated with a carbohydrate chain. The carbohydrate component gives the glycoprotein the glyco prefix. Again, you have these alpha helical or these beta pleated sheet secondary protein structures that can be embedded. You can also have enzymes and other tertiary and quaternary proteins that are more globular in shape that are embedded in as well. Okay, so another thing that isn't really identified in this diagram is that within the cell membrane you also have these little cholesterol molecules which are always oriented with the tails within the hydrophobic zone, and the cholesterol gives the cell membrane the rigidity that gives some structural complexity to the cell membrane. Okay. Again, two main things. One, it separates the cell from the external environment, so it serves as a boundary between the inside and outside of the cell, and it controls the exchange, and that is a semi-permeable process. Okay, it doesn't allow everything to go through. That's the cell membrane. Now, switching over here to the cytoplasm is everything from the membrane including the nucleus, and everything within at least a eukaryotic cell is lipid-based, or organelle-based, meaning a lot of the organelles that we're going to talk about in a minute have their own cell membrane or their own lipid bilayer, And all of the organelles look different. They all have a different structure, and that's really important to note because they all have a different function. And organelles are really good about structuring themselves, or have been structured in a way that allows them to function the way that they're supposed to function. Okay, so cytoplasmic organelles divide to cell into a number of functional compartments. Basically, they divide the cell, that's a typo, it should say divide the cell into a number of functional compartments. Each one of these compartments are called organelles, and all of them have their own structure because they all function independently of each other. So if we zoom in to this particular eukaryotic depiction, or this eukaryotic picture, you'll see a variety of organelles. Again, I'm going to assume that because you're in an advanced biology class that you already understand these, but we're going to kind of talk through the functions of these just as a review. Okay, so we're going to start with the nucleus. The nucleus is this large structure kind of towards the center of the cell. It is a large organelle. It has its own membrane. The membrane is called a nuclear envelope. It is porous. You'll see these little pores, or these little holes that are coming through, and that allows the ribosomes, which are made inside the nucleus, to leave and get kind of attached, or leave and attach to these rough ER. It also allows for mRNA or other RNA molecules to be produced in the nucleus and then leave, obviously, to produce a structure, or at least to produce the process of transcription. But the nucleus as a whole contains the genetic information necessary to control cell structure and function. It has the information, the hereditary information, DNA, chromosomes, all of that is kept within the nucleus. Okay, the nucleolus produces ribosomes. So these ribosomes are going to be made inside the nucleolus, and they're going to literally leave the nucleus through that nuclear envelope, specifically through a pore, and are going to attach themselves to the rough ER. Okay, what do those ribosomes do? The ribosomes are going to aid in the production of proteins on the rough ER and in ribosomal complexes called polysomes. It is important to note, though, that not all ribosomes are going to attach themselves to the rough ER. A large portion of them will, but some ribosomes you will see are going to be out here non-attached or disattached from the nucleus, and these ribosomes are technically going to be attached to cytoskeleton components, specifically microtubules, that are just not really depicted in this very well. Okay, it does show down here that there are some depictions of the microtubules, but those microtubules will be running everywhere on the inside, and so there are obviously in this picture a lot fewer than there actually is. Smooth ER is the endoplasmic reticulum kind of on the exterior of the rough ER. The smooth ER does not possess the ribosomes, which gives it the appearance of being smooth, which is where it got its name. It produces phospholipids and other lipid-based components, as well as stores calcium ions, which is really important for muscle contraction because calcium ions are important for sarcoplasmic, kind of that sarcoplasmic shortening. Okay, so rough ER, I kind of skipped over that. The rough ER is the site of protein synthesis, and specifically, and I'm going to talk about this on the next slide, but because there are ribosomes in two different spots, technically the proteins that are made by the ribosomes in this location are going to be used differently than the proteins that are used or made in this location. Okay, the rough ER, because it is so closely oriented to the Golgi apparatus, the proteins associated with the rough ER are actually more likely to make proteins associated for extracellular use, meaning the proteins that are made by the ribosomes on the rough ER are going to be sent to the Golgi apparatus and then will be sent exteriorly to the cell, meaning they're going to be made and packaged for export, whereas the proteins that are made by the ribosomes on the microtubules that are out in the cytoplasm are technically going to be used in-house, and more on that will come. Okay, the microtubule is the largest cytoskeleton component, and it's used to provide structure, support, transport, and shape on the inside of the cell. It's basically analogous to the largest bones in our body, which provide us shape, structure, and support from obviously the inside of our body. Okay, centrioles are these little guys. They're going to aid in cell division. They produce spindle fibers when the cell is actively dividing, and those spindle fibers not only line up and move chromosomes around, but they also lengthen and shorten the entire cell, leading to the cleavage furrow and then ultimately leading to cell division. Golgi body is going to sit out here kind of exteriorly to the rough ER. It's going to receive protein products from the rough ER and is going to sort, chemically modify, and package those proteins for export, which, like I said, will be sent on to the exterior of the cell. Lysosomes are these little things that are basically vesicles that contain digestive enzymes. They're going to be budding off of the Golgi apparatus and are going to be used in-house. They combine with food vacuoles and help to digest materials that are engulfed by the cell. Basically, those are the trash trucks or the garbage trucks of the cell. They're designed to kind of recycle and degrade a lot of the food and water and pathogenic material that gets into the cell. Mitochondria are the sites of ATP production. So mitochondria would be the site of cellular respiration. And mitochondria are really, really important components of eukaryotic cell because ATP is vital to the sustaining of the cell as a whole. One thing that isn't depicted very well is a secretory vesicle. They are going to help store hormones or enzymes in the cytoplasm, and they just wait for a signal that signals the release of those hormones or enzymes, and then when they get the signal, they will release those hormones and enzymes to usually the exterior or interior of the cell. So we just talked about kind of the route that a protein for export will take, and this depiction is going to be used to kind of illustrate that. We know that ribosomes that are on the surface of this rough ER are going to ultimately be made in the nucleolus. Okay? Once the nucleolus produces the ribosome, it's going to be sent out through one of the pores and is going to embed itself in the membrane of that rough ER. Once that ribosome is in the membrane or embedded in the membrane of that rough ER, it will sit there and help to synthesize amino acid chains, which are proteins. But typically, the rough ER, and more importantly, the ribosomes that are on the surface of this rough ER, are just going to make proteins that are in the primary protein structure, which is just the straight chain of amino acids. And then once they are made, they're going to be kind of sent to the Golgi in the secretory vesicle, where the Golgi will chemically modify and package those proteins into either secretory vesicles or lysosomes, meaning they're either going to stay in or they can actually be excreted out of the cell, which is important for cellular communication. There are, in some cases, though, some genetic disorders, including cystic fibrosis, that has a defective or is the result of a defective protein that don't fold properly within the rough ER and are destroyed, resulting in the clinical symptoms of the disorder, which we're going to talk about cystic fibrosis a lot in this class. Okay, so typically after these protein products are made and slightly folded or kind of initially folded by the rough ER, they're going to be sent to the Golgi, where the Golgi receives them. They're going to be packaged and sorted and chemically modified and folded further and matured, and are going to be delivered to specific destinations both inside and outside of the cell. Okay. Lysosomes, like we talked about in that previous slide, are one of those that are membrane bound, or it's a membrane enclosed vesicle that contains digestive enzymes specifically. They are made in the rough ER and they are used inside and outside of the cell as the garbage truck, which cleans up a lot of foreign materials. Okay, proteins, fats, carbohydrates, and worn out organelles in the cell that are marked for destruction end up in lysosomes, where they can be broken down and recycled so that the cell can reuse those organic molecules and those other kind of molecular components to help produce things that the cell needs. Okay, the last organelle we're going to talk about is the mitochondria. Again, we've already talked about it being the site of energy conversion, meaning it is the site of cellular respiration. Which I know we've talked about before, but cellular respiration is the way that leads to large amounts of ATP being made, and cell respiration helps to turn glucose, which is the energy source, into ATP. Okay, mitochondria contain a circular ring of DNA, which is a plasmid. You can see it here. The cool thing about mitochondrial DNA is that it is independent DNA from your nuclear DNA. And because DNA is kept within the mitochondria, the mitochondria replicate itself, leading to an increased number of mitochondria, which is pretty cool. It's the only organelle that can actually self-replicate, and it's in large part because it contains its own DNA. The mutations in that mitochondrial DNA affect mitochondrial function and cause several genetic disorders as well, which we'll get into in this class. Okay? The nucleus, this is where all of our DNA is kept, and when we talk about DNA, which I know we've done in this class already, but DNA is kept in structures known as chromosomes. Okay? And so the nucleolus is the core where all of those ribosomes are synthesized. And the state of the DNA in non-dividing times is called chromatin. Okay, chromatin can kind of be seen here. It's basically a form of supercoiled DNA that isn't totally supercoiled. It's kind of like first or second level coiled. Chromatin is only going to be supercoiled or supercondensed into chromosomes during times of active cell division. Chromosomes can actually be oriented in a karyotype or in a kind of graph or depiction where all of the chromosomes can be viewed at once. This is really important when you're trying to identify not only gender, but also specific chromosome abnormalities at the chromosome level, okay, which can lead you to a greater understanding of their genetic disorders, potentially. That's where we're going to leave it. If you have any questions, bring them to class. See ya!